Right, let's see. Go. There we go. That's what I wanted. Hi guys, welcome. If you are joining me, if you have any questions, please do ask away. Just setting it all up. Hi, welcome. If you are joining me, if you have questions, please do ask. Hi, Olivia. Right, what are you doing there? And we'll get going here. I'm hoping that this is giving a good sound, but if not, that's fine. What was I wanting to do the other day? I was going to Hi guys, welcome if you are joining me. I am here to answer any questions you may have. If you have any, please do feel free to ask away. And whilst I am waiting, one of the things that I tend to do is I like to make little mind maps to remind myself of what I need to plan when I'm doing my courses. And so it's important to always be looking back on the specification and seeing what it says. So welcome if you are joining me on my YouTube channel. If you are on my TikTok, hi, welcome. As I said, I am just here to answer any questions you might have. Feel free to ask away. That's funny. That's not what I wanted to do. So one of the things that I've been doing today is I have been making notes to myself for the cloning part of the course. So when you are doing some revision, what I would suggest you do is to make mind maps. Basically, I love mind maps. It's one of those things that I find so much I can gain from writing little notes to myself and turning it into my own notes rather than just the rewriting what's in the textbook. So that's one of the things that I would say that I do is I start to look at, well, what is it that is going to help me remember what are my trigger words? Do you think uni is easier than A levels, like healthcare courses specifically? I think that first year of uni is a bit at the beginning, recap, and then taking you further. But again, it depends on where your strengths lie. If you choose a course that you are really interested in and engaged in, and that part of the course you found easier, then yeah, I would say that uni sometimes can be a little bit easier. There's less pressure on you to get to that end point of the final 2 years and to get the grades you need to get into university. When you get to university, it's about just passing the grades to continue. So I would say, yeah, potentially it's a little bit easier. It's better because you're not having to learn. Yeah, absolutely agree on that one. The fact that you're doing the stuff that you enjoy rather than all the topic areas. So when I was at uni, in my final year of university, I had to pick specific modules to do, and one of them was about microbes in the soil. And it's funny now I look back at that, I hated that topic area. I was so bored. I did not enjoy it. But I had to do it. So there are still topic areas in your university course that you'll be like, oh, what is this now? I'm like, oh, actually, that's really quite interesting. I wish I knew more back then when I was doing the course. It depends on the actual exam board and the university as to whether they are multiple choice. So my first year, all my exams were multiple choice questions in the first year, and as long as you passed by a certain level, you didn't have to sit written exams. But if you couldn't pass the level on the multiple choice questions, you had to sit the written exam. Now I'm pretty good at multiple choice questions, so therefore I aced them and didn't have to sit any exams my first year of uni, which was great. Yeah, it's an interesting one. A levels are specific, and depending on the exam board, it's specific what they want you to know and what they're going to assess you on. So I would agree with you on that one. A levels do have specific mark schemes, and universities are more. It's down to your lecturers and their professional judgment on what grade. Yes, they have criteria, but it is like your 6 markers in the exams. The 6 marker that you do, we read it and we give a professional judgment of where you sit within that criteria, but we have guidance, they do too. But again, it's about the universities and who's actually marking it and what they will accept, what they won't accept. It's a fascinating world, basically. For the most part, I think yeah, it's about being able to get through the course to the extent of what's needed, but then you actually become an expert later on. It's not about the first couple of years. The first couple of years is making sure you've got the skills leading up to it. See, the thing is with the answers, you said whereas in A levels you can have the knowledge but get a C. It's not about having the knowledge, and that's where a lot of students fall down. They think they just have to learn the content, but that's not what it's about. It's not what any science exam is about. It's about you having the knowledge and then you being able to use that and apply it to the different context that they give it to you. So it depends what they want to assess you on, but if you have the foundational knowledge and you are able to apply it to the question, you shouldn't be getting C's. I would definitely say that you need to make sure that you are working at your written answers if you're still getting C's when you feel like you know the content. The other thing I would also say is you feel like you know the content, but do you actually know it, or do you surface level know it? And I mean that in a nice possible way, because a lot of students say to me, "Oh yeah, I know this, Christine." And when I pick apart and I ask them questions, they're like, "Oh, maybe I didn't know that." So, for example, I was doing a session tonight and we were talking about PCR. Hi, and we were talking about um, a multiple choice question, and the multiple choice question said about the DNA and it being positively or negatively charged. And I asked the question as to why is it negatively charged, and they knew it was negatively charged and they could answer the question that was in front of them. But when I prodded and asked the question as to, well, why is it negatively charged, can you tell me that part of it, that little bit of information was missing from the knowledge. And because that was missing, it meant if that question wasn't a multiple choice question where they were able to get the right answer because it was in front of them, and instead it had been switched to a 2-3 marker, they could have lost those marks and then not got the grade that they wanted because they didn't have that little bit of information that they needed. And it's making sure that you don't just know the content in the sense that you've memorized the content. Can you then apply what you've been doing to the next part? So another question we were looking at was to do with PCR, and it was about being able to know how many DNA fragments would you have after, let's say, 12 cycles of the PCR machine. So if you've got 12 cycles of the PCR machine and you are starting off with one DNA, imagine you've got one DNA fragment, how many would you have at the end? And then they got them to do it in the answer had to be in log 10. And so that kind of skill, do you know how to use your calculator to switch from the number? Do you know how to calculate a number? So obviously, because it's semi-conservative replication, because you have it going from 1 to 2 every cycle, you therefore would have 2 to the power of however many cycles. So in this case, it was 2 to the power of 12. So we had 4,000, I think, and 96 DNA fragments. And then if you turn that into a logarithmic scale, you can then note that that's 10 to the power of 3.61. So practicing those sorts of skills and understanding how to use that in the exam, that's the sort of thing you need to be working on. If, again, going back to what you said originally, you feel like you know the content, but at A levels you're still only getting C grades, that would be one of the reasons potentially that's holding you back. Do you have the detail in your knowledge in your answer, should I say, sorry, to get to that higher level? Or do you have limited understanding because you've memorized the content? And memorizing the content does not mean you know it. And that would be where a lot of students struggle, which is a bit of a shame. But that's hopefully where my videos are helping you to get that. Your live sign came up on my phone. Oh, earlier this year when you were revising for your exams. Oh, thank you so much. That's lovely to hear. It's a funny one. That I have so many students that I have worked with over the years, and it's really lovely when they pop in and they say, come and say hi to me, and that I've worked with them. And it's weird to think how many students remember me and my teaching, and I'm a part of their story. So I was saying this in my live the other week there, and it's very relevant because our car is back in the garage again. But the person who works in the garage is the salesperson in the garage. Actually, is a person who I taught. How many years ago now? Must have been about 2010. They were in year 7 and I was their science teacher, which is really quite funny. And they remembered me. And when I turned up to put my car in, they basically turned around and said, "Are you a teacher?" and did you teach science? And you taught me when I was in year 7. I was like, oh my gosh. And it was an old school as well. Um, Thank you for all your resources. Guys, please use our videos, especially since they aren't many. Oh, resource for OCR A. Thank you. That's really lovely. I have been building up more and more resources and putting it on there. So the whole course is there. So if you finished your exam, you would have been where I was building it slowly up. Whereas now the whole course is there, so students have access to everything that they need. And I'm building the resources moving forward for my February. I'm hoping it'll be done by February revision course, if not by the Easter one. And I'm always improving it. Tips for revising your PAGs. Your PAGs are not really something you can revise, so don't stress out about the PAGs that you're doing. Okay, the key thing about the PAGs is about the skills. So you'll be assessed on your skills rather than the PAG itself. So you might have done one PAG, whereas another teacher does another PAG, and that's okay, that's not a problem. OCR don't set required practicals, they set PAGs 1 to 12, and teachers have the freedom to do whichever they want to as long as they have assessed you on your CPAC skills. So the key thing for revising a PAG, to pass the PAGs, follow the instructions, draw your graphs, do what is needed safely, using the equipment that's there in the right way. So these are things that you should be failing some of your little parts that you're being assessed on in your PAGs and showing progression. So if you're passing them all straight away, that actually looks really bad on the school, that you're not showing any progression whatsoever. So don't stress out if you've missed one or two of the skills in the PAG so far, because your teacher should be giving you enough opportunities to pick that up, so you should get the endorsement at the end. So obviously they're not going to fake it, they're not going to say that you're passing it when you're not. They're going to give you the opportunities to get there. However, what I would say is practicing for the exams. So do you know how to draw a conclusion? Do you know how to evaluate? Do you know what type of variables should be there in a practical? So that, I would say, is things that you can practice. You can use your past papers to do lots of practice questions and try that as best you can. But revising for the PAGs isn't really revising the PAGs. You should be revising the specification and all the practical skills that are dotted throughout the specification. So if you're looking at the specification, then you should see where it says there's a PAG 1, there's a PAG 2, and it comes up in lots of different places. This one could be a PAG 3 or a PAG 5, that kind of thing. So if it is potentially going to be assessed, it could be anywhere within your specification. What are high yield topics for paper 1? So paper 1 is obviously modules 1, 2, 3 and 5. So therefore on that one, it depends on which year and what they assess. I've actually gone through and I've analysed every paper from 2017 right the way through to 2025. And each year they do different ones. So if it's in a multiple choice section in one year, it will then become part of a section B in the next year. So there isn't a high yield topic for paper 1 because they cycle through them all. So the one thing I would say to you when you're revising the topics is module 3 and 5, you should be revising them, and topic 2 should be coming in all the way through all of those topic areas, but you're never going to know which way round they're going to ask the questions, because obviously that's the whole point. You shouldn't be able to predict. I will do this is my best guess, but what I would say is you need to know all, because you don't know, and I don't know which way they're going to go. I can make assumptions, I can make suggestions, but I don't know. I don't work for the exam board. I'm not the one who's writing the exam papers. One thing I will say is when you look at the papers, when you look at the trends, because that's what we do as scientists, we look at patterns, we look at trends, you can then work out where they may go with some questions, would be what I say. Your videos got me an A in my December mocks. That's amazing. Congratulations. That's really, really amazing. And I'm so glad that I could be a part of that journey to help you. So happy. Thank you for sharing that with me. What is rigors the fever and why does one shake when it happens? Do you happen to know? Okay, so when it comes to fever, that's an interesting one. When you get a fever is where you are getting a change in your thermostat, I would say, that's inside your body. So right now it is really cold outside. So in my house, I have my heating on. I've got a thermostat that's set that says, right, okay, when the temperature gets to this point, when it gets too low, switch on the heater, switch on the boiler. We're going to start heating the room up. When it gets to a set level, we're going to switch off all the heating. We're now at a nice comfortable temperature. Switch it off, and therefore it will be at a comfortable position. Okay, so I'm using heating as an example because it's cold outside. So if you think about that, and then we're talking about our internal set levels. So we have got a set level. We have thermoreceptors in our hypothalamus, which are detecting the temperature of our blood as it circulates around. When our blood temperature gets too low, when there is a decrease, that is going to be detected by our receptors. Our receptors in our hypothalamus are then going to activate our thermoregulatory controls to heat us back up again. So when we get too cold, we are going to have a set of programs that are going to work in our body to bring the temperature back up. So if I'm too cold, I'm going to have impulses sent to my muscles, telling my muscles to contract, and that is going to cause me to shiver. And I'm going to have these fast contractions. I'm shivering, my body, my teeth might start to chatter because I've got my muscles contracting really quickly. I am trying to generate heat. I've got my pilorectal muscles are going to contract, which makes my hair stand up, trapping a layer of air. And I'm going to have increased respiration occurring so that I am generating more heat. So the key thing that's going to happen is I'm going to try to reverse the drop in my blood temperature by all these physiological processes happening. So that's a thing that I do because I'm an endotherm. Now, with a fever, when someone has a fever, what happens is that bottom end temperature where your body has signaled, the receptor has detected that the temperature is getting too low, we need to switch on the heating processes, the heating the body up, that level has changed and got a bit higher. And so because of that, it means you don't have to have a bigger drop before your body thinks it's cold and it starts to respond in the same way. So you are shaking even though your body is really hot because your thermostat, your set level, has been pushed upwards. And that's a way of positive feedback because you want to increase the body temperature to slow down the reproduction of the pathogen. So it's a way of your body responding to an invader, getting that temperature hot so that it's affecting the proteins, denaturation. If it goes too high, it becomes a real problem for us, but it will slow the rate of reproduction down. And that's why you get that shivering side of things. Hi Chidima, how are you? So I'm just trying to keep up on the questions that are going. If I've missed any, I do apologise. Miss Sid, I just saw you're in here. How are you doing? Is this A-level? Yes, it is A-level. Please explain process of breathing. Okay, so your breathing is actually a really easy one if, again, we go back to thinking about muscles. So you have got your rib cage, and you have got your intercostal muscles, which are between your rib cage. So that's the first thing we're going to talk about, the intercostal muscles. The other thing we're going to talk about is our diaphragm. So basically what we have, so imagine, here we are, this is my thorax, and inside here I have got my lungs. Okay, so my lungs are attached to my arms here. Now, when my intercostal muscles, and it's important you note the external intercostal muscles, when the external intercostal muscles contract, they are going to pull my ribs upwards and outwards. Now, if my lungs are attached to my arms, they're going to be pulled along with my muscles. And because they're being pulled along with my muscles, what's going to happen is that's going to result in a larger volume within my thorax. So you can see as my ribs are going up and out, I've now got a lot more space in between. So because I've got a lot more space here, my volume has increased. And because my volume has increased, my pressure has decreased. That's going to mean that the air is going to flood into my lungs because it's moving in down a pressure gradient. Now, what I need to understand, and this is the bit that students get wrong, when my muscles are actually, I'm going to do this here. I'm going to use my bubble. Let's take out my hair. It's been in a plait all day. Gonna have a bit of a wave to it. There we go. There's a bit of a wave for you. But having a bubble in your hair is always good for you. So here we have, this is my alveoli. Okay, so here is, or I should say alveolus because it's singular. Here's my alveolus. And what I'm going to do is it has elastic fiber. So you can see that it's open. I'm going to stick my hands in to represent what happens. So when my external intercostal muscles contract, when they pull the ribs up and out, the lungs are going to go with them. And as the lungs go with them, they are going to stretch. So as I'm doing with my bubble, I'm stretching it. And what will happen is I've now got more volume inside here. I've therefore got a lower pressure. So all my alveoli are doing this at the same time because they're being pulled up with the ribs going up and out. They are therefore stretching, they're opening out, the volume is changing inside. Now, this is pulling me back in. It's really quite difficult. This is like working my muscles by pulling this apart. So when your ribs are being pulled up and out by your muscles contracting, they are stretching the alveoli. And because they're stretching the alveoli, what's going to happen is that's going to result in the elastic fibers, when those muscles stop contracting, so when the external intercostal muscles stop contracting, the elastic fibers are going to be able to recoil. And they're going to recoil, bringing the alveoli back to the original position that they were in. So therefore, we have this stretch, changing the volume, and this recoil, bringing it back in. Now, because it's gone from a larger area to now being a smaller, so the volume has decreased, the pressure has now increased, that's going to force the air out of the lungs because we've got a higher pressure now than the outside. So it's all to do with those external intercostal muscles and the fact that they're contracting, lifting the rib cage up and out. Now, I also mentioned about my diaphragm, which is down here. So the rib cage comes up and out. My diaphragm was a dome shape. When it contracts, it drops down. Again, a bigger volume. So when I am going to relax that, what's going to happen is that's going to relax, come back up to the dome shape. My muscles are going to relax, and that's going to pull me into a original position that I was in. Now, if I go and I really force out the breath, I'm using different muscles this time. So that forcing out of the breath that I just did over my tidal volume, that is where my internal intercostal muscles, so I've got muscles on the inside, internal intercostal muscles, what they're going to do is they're going to contract a little bit further. Because they're contracting further, they're going to pull the rib cage down and inwards. That's forced expiration. So that's different to the normal breathing that's happening by the external intercostal muscles and the elastic fibers. The forcing, that pulling in even further, that's the internal intercostal muscles. But you never fully actually change that doesn't make sense. You never fully actually empty your lungs. You're always left with a residual volume inside. So it's basically like, I can try and squeeze this together, but do you see how I'm still getting these bits that are not quite fully being squashed? Well, that's the same with the lungs. When you are forcing the expiration, you never fully collapse the lungs. You don't want to fully collapse the lungs. So you've still got a little bit of stale air, which is left inside your lungs, and that's your residual volume. So when you're doing the spirometer and you're looking at it, should be able to work out your vital capacity, your tidal volume, your inspiry reserve volume, your expiry reserve volume, and your residual volume based on the information given in a graph. Oh, no, didn't want to do that. I'll go away, go back up. Not to you guys, I hate when I do this. Still getting used to my phone, and so therefore, I can't slide you back up now, I can't see. Oh, how have I just made myself so, oh no, oh, that's gone even worse. That's so weird. There was a question there. I was trying to read a question and it totally went. Where'd the question go? Is it correct to say the thoracic volume changes, changing the pulmonary pressure? So yes, you want to talk about the thoracic volume changing, and that therefore would change the pressure. I would be a bit hesitant to add in about the thoracic and pulmonary. Not hesitant. Obviously, you're going into a bit more detail, but in the A level, you don't have to go to that level of detail. What you want to do is talk about volume changing and pressure changing. And the reason I say that is because they might not ask you about, hi, sorry, I've just seen someone's asking me questions on YouTube. Give me 1 second. I'm just going to finish this question. What I would say to you on the talking about the volume and the pressure, the reason I'm saying to just keep to those words is because if we're talking about fish and we were talking about the way in which the exchange happens in fish, the buccal cavity opening up, therefore the volume changing, therefore the pressure changing. So I would be using volume and pressure and making sure I've got that, and then applying it to the question that they're asking you in the exam. You are very welcome, Joko22. Very, very welcome. Thank you for the likes. Right, hold on. What have we got here? I'll put, Miss, can you do 6 markers in AQA combined biology? So you're talking AQA combined biology would be GCSE level. I'm actually doing the A level sessions is what I do just now. I will be back probably not on the 5th, probably the week after. I will be back and doing some more GCSEs again. What topic area are you struggling with the most when it comes to, for example, a 6 marker? Because the combined science can go in lots of different ways, but I wouldn't say that, just done it again, that they are ones to worry about. Chidima, I am coming to you. I'm sorry, I did see your question before, but I wanted to finish that. So revise basically the whole, and realize I need to chill as the holidays isn't over. Yes, enjoy the holidays. Question on cell mediated response, right? Ian, give me a second. I'll come back to mitosis and meiosis after I've done this one from Chidima. Is the binding of the T cell receptor to the antigen on the APC clonal selection? So yes, you would have, so the T cell receptor is a really interesting one, because I have a friend of mine who works in T cell recognition, and I keep picking his brain more and more about it. So the T cell receptor and the antigen on looking at the APC clonal selection, yes it is. So clonal selection is selecting the correct cell that needs to be cloned. So if you've got a T cell, that T cell needs to be cloned to therefore then differentiate into whether you've got your T helper cell, your T killer cells, whether you've got your memory cells. So what you have to understand is the recognition of an antigen as being foreign when it has been presented by an antigen presenting cell. That T cell going through a form of mitosis, that is clonal selection. You've selected the correct T cell that is therefore going to go through the process of mitotic division. So clonal selection is selecting the right cell that is therefore going to be copied to produce genetically identical cells, which therefore results in you having lots of them that can then differentiate into whichever cells they need to. Do you need a T helper cell for clonal selection? So if we're talking about the B cell, so it's an interesting one because clonal selection could be the T cells or it could be the B cells, depending on where we're going with our question. So coming back to this, you would need the T helper cell for the B cell to be activated to go through that process of clonal expansion. So clonal selection is to do with the antibody being produced. Hold on, let me take a step back. Clonal selection is where you've got the antigen being recognized by the B cell and the T helper cell fully activating the B cell to go through clonal expansion. So clonal selection is selecting the right B cell to go through the clonal expansion. So it's a difficult one because it depends where you're starting the process from, of whether it's clonal selection or whether it's clonal expansion and which one. So you're selecting the right cell. So for OCR, if they're talking about clonal selection, they're pretty much talking about the B cell that's going to make the right antibody against the antigen being selected, and then the clonal expansion occurs because you've got your T helper cell fully activating the B cell to go through the clonal expansion. I hope that helps and made sense. Let me just double check what else we've got here. So you had mitosis and meiosis. Thank you. Does the B cell present the antigen in your... The B cell can present the antigen, yes, on its MHC complex. It's an interesting one. You want to limit the response that you've got. You don't want it to be too big a response, because too many T cells, for example, could actually cause a problem within your body, because you could start to damage cells that are not needing to be damaged. And so therefore, it's really interesting. There's a lot more detail that you have to go into if you study this later on, but you have your macrophages that are going to obviously engulf, digest, and then present on their cells as being the antigen presenting cells. But you need the T cell to actually also be activated before it can then start to produce the cytokines to then activate the B cells. So it's such a more complex thing than you need to know when it comes to the actual process. But it's important that when you are doing and talking about it, you talk about the selection of the correct cell, the expansion of the cells, and whether it's a helper cell that's making interleukins or cytokines, or whether it's a killer cell that's making the perforin or the hydrogen peroxide, whatever it is. Each of the different cells have got different roles to play, and so yeah, it's really interesting. And it is a difficult concept to get. The key thing is knowing that you are answering the question that they've given you in the exam, not just memorizing the content. So what example do? I am currently doing OCR A. I have a student, actually, shout out to the student, bless them. They keep asking me about AQA group tuition. So I've built all my resources for OCR A because there wasn't nothing out there when I started 3 years ago for, was it 3 years ago? October 2022 was when I started teaching OCR again. I hadn't done it for a while. So I went looking for resources to direct my students to, and there was very little out there. And so because there was very little out there, I started producing resources for my students. And then I started doing the TikToks to promote my revision platform, actually, that I did, aiqchat.com. So I was promoting that based on a student suggestion, and then they then said to me, why don't you do TikTok? So I did TikTok, and then whilst I was doing TikTok, they in 2023, I think it was It was on the day of the first exam. I started going live on TikTok, and they then said, do you have a YouTube channel? I was like, well, no. So then I went to the YouTube channel, and I built the YouTube channel up in 24 to 25. And then, yeah, I've been doing it again, 25, 26. Now, Ellie, what I would say to you is I tutored a student last year on Edexcel B, and I would use my resources for the sessions that I did with them, because a lot of the content overlaps. There's like a 75% overlap in all the exam boards, but it's just about making sure it goes into the detail specific for the exam that you are doing. So you need to use your specification alongside. So for example, OCR, I was talking about cloning today, whereas there isn't that in other exam boards. So it's just about what do you need to know and what is relevant, what's not. Could you explain end product inhibition, please? Yeah, of course I can. What is end product? Right, end product inhibition is end product inhibition. End product is an end product that is going to inhibit somewhere in the pathway that's earlier on to prevent it being made. So how would I describe end product inhibition? So think about it in this way. Imagine that you are sitting, here I am in this office, I have a printer there, and my role is that I need to print out a whole load of letters, put them into an envelope, and then post them. Now, if my printer is printing out the letters, that's fine, absolutely fine. I then take the letter, I put it in the envelope, I then finish it, I post it. Take the next one, finish it, post it. Now, what will happen is I will end up with a big pile of end product here. The end product here is getting too much. I've got too much. I just have got too many end products in this room. It is overwhelming. It's too much. I'm going to stop the printer for a little while. I'm going to stop the letters coming out because I need to deal with this. I need to get to the post office. I need to get rid of the letters because there's just too much. So if there is too much of an end product, that end product, as that concentration rises, that then has a knock-on effect on the production of the end product, and it depends where it's going to have a knock-on effect. So because I am on my channel, I will put it up here, because if anyone comes to it, all they've been looking at is vegetative propagation. So what do I mean? I've got A is going to become B, B is going to become C, C is going to become D, D is going to become E, E is going to become F. Right, so I have got enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. So I have my substrate A, which is going to be broken down. We have an enzyme number 1, which is going to obviously catalyze the reaction, speed up the rate of the reaction, and that's going to result in us getting, our product, which is B. B is the substrate for enzyme 2, which is going to result in my product of C. C is the substrate for 3, and so forth. So I have these intermediate products. Now, what I have to understand is F is my end product. So you said about end product inhibition. I have my end product. My end product is going to potentially stop enzyme 1, or it might stop enzyme 2, or it might stop enzyme 3, it may stop enzyme 4, it may even stop enzyme 5. It's an end product that is going to inhibit the pathway in the production of itself. So because we are inhibiting it, it's straight away we know going to be a reversible inhibition. We don't want it to be permanent. So we know it's a reversible inhibitor. We know it's the end product, so it's not going to compete. It's going to go for the allosteric site. And what it's going to do is it's going to change the tertiary structure of the enzyme, therefore affecting the active site shape. And it affects the active site shape for a short amount of time whilst it gets rid of the products. So the products go off and are used elsewhere, and then the pathway can start again. So it's an end product inhibition because it's the end product which is going to stop the production of itself until the cells can deal with whatever it is that may be. I hope that answers your question. And that helps on that one. Thank you, by the way, for the likes, for the people who are sharing and liking it. That is pretty much what I do these sessions for. I do them so I can answer the questions that you may have to help you, because not everyone I know can afford tuition. Not everyone can afford to pay someone like me with experience. And that's the one thing I would say to you. If you are looking for a tutor out there, please do make sure that you are kind of vetting them to see, are they a good fit for you? I will not be the best tutor for every student out there. I am very unique in the way that I teach. I teach the way I teach because it's how my brain works. And for some of you, that might not be the right thing. But for others, it does work because it is breaking it down. It's finding those links. And I want to help as much as I can. But obviously, I have to prioritize the time that I can do and when I can't. So I have a student who is wishing for AQA group tuition sessions, which I am tempted to do, but I need 3 students at least for me to offer it out for a cheaper price, obviously. And at the moment, unfortunately, this one student is the only one who's looking for it at the moment. And I get it, because a lot of people are like, well, you do OCR, and I do OCR because I've made a point of making sure I know that spec inside out. I do tutor AQA. I have that a lot of one-to-one clients on that front, but the group tuition side of things is very different, because when you're in a group situation, you've been taught different things. So I need to make sure that everybody is able to get the resources to what they need to. And so, yeah, for the group tuition to run, I would need at least 3. So if anyone is AQA and is looking for a reduced cost group tuition session, then please do send me an email. I wasn't sure about streaming today, to be honest. I was doing a session for a student and I jumped on. I was on last night, but I decided to do a little bit because I'm here, I can do it. And my brain is very active at the moment for the course that I'm going to be running in February and Easter. I'm hoping it'll be done by February. If not, it'll be done by Easter. But yeah, I thought I would jump on and see if anyone needed any help. That, and because I've been very late in going to bed lately, and so my brain is very active at the moment. And also it's quite nice to not just be dealing with a 5-year-old and a 6-year-old. I love my kids to bits. I do. I love them to bits. But it is very exhausting. And this is my passion. So for those of you who don't know me, I obviously have ADHD and potentially a little bit of autism. And so therefore, what I would say, I say a little bit, I have autistic traits, definitely. I can be very strict on certain things. And yeah. Anyway, distraction away. What I would say is, for me, this is where my interest is. This is where my passions are with biology. And so I like doing this. I'm always thinking about biology. I'm always thinking about how I can make more videos. I wish I had the space and time to be able to do more, because I have so many ideas. I have so many books with like little notes and little thoughts of what I'm doing everywhere, but unfortunately time gets away from me. So I do these sessions so that I can help. I can answer questions, I can offer what I can do, and then when I am fully in my sessions with my students, I have planned them to the nth degree, and they are definitely getting their money's worth, I should say. So yeah, so I do offer these wherever I can. What are we doing today? Whatever people are asking, basically. Any questions you have, I do try and answer. But I will be back officially, again, after the festive periods are over, I'll be back on a Wednesday as a normal session, basically. So Wednesday evenings will be my normal sessions that I do. So yeah, that will be the norm. But if you have a question, please ask away. Excuse me. But right now I'm just chatting away, answering anything that anyone has. Oh, that's not even showing the end of my screen. How bizarre. See, that goes to show I wasn't actually looking at that properly earlier. But I am only going to stay on for probably about 5 more minutes because I have a few emails I need to respond to. And I need to get on and get myself ready for my new sessions. Rajveer, you don't get DNA. What part of DNA do you not get? What bit are you struggling with? So if we talk about DNA, what bit of the DNA? So what tips can I give you when you're writing 6 markers? So the 6 markers, what I would say, if I'm going to give you any tips on that one, is look at the question. So the questions will normally have potentially 1, 2, or 3 parts of the question. So the 6 markers are always very much about you taking your time reading the questions, looking at what they've actually given you, and then formulating your answer to be right. Well, here's my answer to number 1, here's my answer to number 2, here's my answer to number 3. A lot of students, when they read the questions, tend to only see the third part of the question for some reason. So if I give an example of, I'm trying to think of one off the top of my head. Let's say it's about genes, about mutations, and about proteins. So let's say it's about how the genetic and environment have an effect on a phenotype. I'm just making this up, by the way. So if we're looking at it, I would say, right, well, what is the genetic control of someone's phenotype? So what things are we looking at when we talk about genetic control of the phenotype? So what is your alleles that you possess? Are they dominant? Are they recessive? Going to basics and talking about the genetic side of things, the DNA sequence and how different that may be. Then how could the environment have an effect on the genetic expression? And so how does that then affect the phenotype? And so it's about deciphering the question, taking your time on those questions and saying, right, well, what is 1, what is 2, what is 3? And if they've given you a stem of a question, where does that link into whatever they've asked you? And so that would be my advice when you are working through the questions. Don't just jump to the marks game. Don't just read what they were looking for. Actually dissect that question, dissect the stem of the question, figure out where they have given hints, because there's always hints in the question, always hints. But for some reason, a lot of students don't see those hints. Okay, so I'll finish on this quick one. So you said you don't get the structure of DNA. So if you're not getting the structure of DNA, if you don't understand that structure, what is it that you're looking at? Well, the important thing is whenever we talk about DNA, the first thing we want to start with is a nucleotide. So here's my nucleotide. Now, here is my nitrogenous base. always important to talk about your nitrogenous base. Then we have my pentose sugar, which is my pentose sugar, we know is a number 1, 2, 3, 4, 5 carbon sugar. So if I've got a 5 carbons, there's carbon number 1, there's carbon number 2, there's carbon number 3, there's carbon number 4, there's carbon number 5. I have a pentose sugar, and it's dependent on what is it carbon number 2. If it's a hydrogen, it's a deoxyribose. If it is at carbon number 2, a hydroxyl group, then it is a ribose sugar. So when we look at the DNA structure, we're looking at a deoxyribose sugar because at carbon number 2, there is a hydrogen rather than a hydroxyl group. Christine, have there been enough people who have joined your AQA class? No, I'm afraid not at the moment. I have one person who is really desperate for it to go ahead. I have been talking about it tonight, but unfortunately, because I haven't got the minimum of 3, it's not occurring yet. But if you are doing AQA and you are interested, then please do get in contact, because I will run the course if I can get at least 3 people interested. And I will use only AQA questions, and I will make sure it's very specific to the specification that you guys have. The problem I have is I need at least 3 people to make it viable for me to run, because it's not just about the hour that I'm doing. So right now I'm just chatting away to you, and I am talking about the different parts. I'm covering different parts of the content, but when I actually plan my sessions, so for my group sessions, when you turn up, it is an hour intensive session with me that I've planned. I have resources for you, I've got questions for you, I'm going to show you the application skills. There's a lot that goes on behind in the background that you don't see in preparation for the group session. So for them, I need a minimum of 3 people to make it viable. Otherwise, it's not viable for me to make it work. For the amount of time I put in means I obviously need to block out some time, which means I can't have a one-to-one student on that time, and so forth. And so at the moment, there isn't enough people. I have one person who is desperate for it, and I want to run it for them, but at the moment I can't. I've got an idea that I'm thinking about doing, but I'll see, because one of the ways I could do it is actually have it where I offer the session at an even cheaper cost, but then I live stream it. But they're getting the questions. I don't speak to anyone else. I don't answer any other questions, but I don't know. I need to figure that one out, whether I'm going to run it that way or not. But at the moment, for AQA, I have one person who is desperate for it. I need 2 more, and then I will absolutely run it from the 5th of January. My OCR sessions, if you're OCR, they are running, they are planned, they are ready and raring to go. Because obviously I have enough students in the session to keep them going. So yeah, so if you are interested in OCR, and the groups, the maximum I will go up to in any of my groups is 9, because I don't believe the class should be any more than that, because obviously I need to give you feedback, I need to help you, and so I assess your written ability in my sessions. I am a trained teacher. I've taught classes of 35, I've actually taught classes of 90 to 100 students in large halls. So do you think you could get an A star in the A-level Biology OCR? Honestly, no, I don't. And the reason it's not because I don't know the content. That's not why I don't think I would get an A star. I mean, I probably could get an A star, but I don't think I would, because I trip myself up when it comes to the questions. Although I don't know, because now if I were to do the exams now, I would get the extra time, which would be the processing time that I would need. So actually, yes, I probably could get these. It's a hard one, because when I sat the exam pressure for me is a bit too overwhelming. I know the content inside out, and so therefore I should be able to get it. But what trips me up is that sometimes my brain goes too fast under those pressured conditions, that I sometimes would make a few silly mistakes potentially. So I would hope that I would get the A star. I mean, when I do the papers, but it's not the same as when you're actually sitting it for real. What kinds of pupils get A stars in the exam? It depends. It really depends. There are those students who just know how to answer questions and are able to ace through. There are other students who do the work and work extremely hard and are able to do it. And there are those students who, yeah, just blink and they manage to get it straight away. I don't know. It just depends. There isn't one student, I would say, and there shouldn't be. It's about how you approach it and what you do. I have tutored thousands of students. I have worked with thousands of students, and I have helped a lot of students get the A stars. I've helped a lot of students get the C's. I've helped a lot of students get the E's. It depends on the student. It depends on the individual. I feel like everybody goes, oh, you need to get the A star. No, you don't. You need to get what you need to get to where you want to go. And yes, okay, A star is where you should all aim for. Absolutely 100% aim for the A star. But it doesn't necessarily mean that you need to get the A star. And the reason I say aim for the A star is you shouldn't hold yourself back. If you say to yourself, I'm only a B grade candidate, you actually limit yourself with your brain because you stop yourself pushing past that grade. And so I would say aim for the A star. Absolutely, every single student should aim for the A star. Will it mean that you get it? It just depends. It depends on the questions. It depends on your ability to decipher. It depends on your detail that you put in your answer. So it's really hard to know. Do you do GCSE advice too? Yes, of course I do. I mainly aim at A level now. But I was actually a director of student progress for a school which was all the way up to GCSE, and then I became director of student progress for the sixth form. And I can do it all. It's about where I enjoy it, and that's why I aim mainly at the A level. But with regards to GCSE, I'm putting out videos on GCSE because I'm realizing a lot of students that are coming through into A level don't have the foundationals. They've got the grades to do A level, but they don't have the skills or the knowledge. They have memorized and been spoon fed the content, but they don't understand it. And so what I'm trying to do is I'm trying to help by putting out more videos for GCSE level to help the students who can then bring that into the A level, because A level is hard. It's not an easy topic to, or an easy subject to learn. A lot of students think it is because they like it. They're going to be able to do it, but it's not. Unfortunately, it's quite difficult and quite challenging. But it is 10 o'clock, so I am going to stop now because I need a break. You need a break. You need to go to bed. I have not slept very much last night. My daughters at the moment love a 3 a.m. wake up, which is just amazing. So I'm hoping that I will have a better night's sleep tonight. And I want to say thank you to those of you who have joined me. Thank you for asking the questions. I will be back, not Wednesday this week. I'm not doing Wednesday this week, but next week I will be back to doing my Wednesday evenings. Actually, of course not Wednesday, because Wednesday is Hogmanay. So happy Hogmanay for those of you who are Scottish. Happy New Year's Eve for those of you who are English. And enjoy your last couple of days of 2025, because when you get into 2026 you are going to blink and we are going to be at the exams. There are exactly 20 weeks from the 5th of January, from the Monday, 20 weeks right the way up until the May half term, because I've planned them out for my group tuition courses. And so therefore know that there are 20 weeks of tuition that you could have before you actually get to that exam. And some of the A levels obviously start before the May half term. So some of your A levels are actually in less than 20 weeks. That's scary. But it's also exciting because then you get to the point of you'll be done. once you start, you then finish, and it's then just a waiting game. So thank you for joining me, and I hope you have enjoyed the session. I will be back in the new year, 2026.